So a warm afternoon to all of you, and we are extremely happy that we have all assembled here in good number once again. And looking at the response, we are extremely happy that Copy with Curiosity has been able to deliver the objectives with which it was started. So thank you very much for coming. So I would like to welcome you all on behalf of Jawaharlal Nehru Planetarium to this program by Dr. Shannon Olson. She's from NCBS, and in a short while there will be an introduction of the speaker. So before that, I request Dr. Rajesh Gopakumar, the director of ICTS, to introduce some of the activities of ICTS, and then the speaker by Professor Ramagod.
Thank you, Dr. Madhusudan. So, as always, it's great to see so many of you here on a Sunday afternoon for the Copy with Curiosity event. As many of you know, many of you probably are regulars. This has been going on for a year and a half, and we are always thrilled with the response, and it has steadily been growing now. We even have an online, I think, presence. The live streaming events get a lot of response and even questions sometimes we'll probably see today as well. ICTS, we started this as our flagship outreach event because we felt the importance of communicating the excitement of science to the wider public in Bangalore, and we thought of doing it in the center of town so that people would come in on a, like Vijay says, on a just like you have a coffee, you just come and hear a lecture, and so you get the copy as well as the curiosity. So we've covered a variety of topics from things in black holes to the extremely small, all the way to the extremely complex, and today's will be one on the complexity frontier in a sense of science. ICTS, of course, does outreach, and we have a lot of public lectures even on our campus in Hesarghatta, and do come for some of those as well. But we are primarily a scientific research institution. We do research in areas in mathematics, theoretical physics, and even quantitative biology, and going forward, probably theoretical computer science and related disciplines. But more uniquely, what ICTS does is also have programs by a number of people from a variety of programs on a number of topics in many of the areas that I mentioned. And these are international programs in which some of the best scientists in the world come and interact with the Indian scientific community on topics which are the cutting edge of science today, and many of the speakers in the Copy with Curiosity have been from some of these programs, but we also have people, and today's is an instance where we have from our sister institution, NCBS. So we are NCBS and ICTS are part of the TIFR family, the Tata Institute family. So in short, ICTS is about programs, research, and our outreach. Do look at our webpage. All the talks of the past are also archived there, as well as many other public lectures and other events. And if you are young people who want to learn more about some of the topics in science, go a little bit more into depth. We also have many very good introductory lectures at a more technical level in some of the exciting topics of science today. So check out our YouTube channel, and I think you'll have a lot of stuff there to both entertain and educate you. So with that, let me just stop and hand it over, I think, to Rukmini, who will be introducing the speaker. So welcome again to Shannon. Thanks for agreeing to be the speaker today. And I forgot to also thank the Planetarium, as always, for its hospitality and for partnering in this, and which is why we I think we've had a very long and very sort of a great partnership, and hopefully this will continue. Okay, thank you very much.
Hello, everyone. It's a great pleasure today to welcome Professor Shannon Olson. She heads the Naturalist-Inspired Chemical Ecology Group at NCBS TIFR. This group, called NICE, is particularly interested in how animals, especially insects, identify objects across different environments. Dr. Olson completed her PhD at Cornell University in neurology and behavior and chemical ecology. She then worked at Max Planck Institute for Chemical Ecology in Germany as a project leader, where she collaborated with engineers to develop an artificial chemical communication system based on insect chemical communication. A Fulbright scholar and a Ramanujan fellow, Dr. Olson's favorite thing about being a scientist is the privilege to observe and learn every day some completely new and the possibility that something new could make a positive difference in this world. So we have a lot to learn from her today, and she'll be talking about endless forms, most beautiful. But before she starts, I'd like to invite Rajesh to hand over her memento to her.
Good afternoon, everyone. It's a great honor and a privilege to be here. Thank you to my colleagues at ICTS and also to the wonderful planetarium for having this program. I've been following it for several months now, watching the different talks, so I know how much of an honor it is to be invited to speak. And today I'm going to tell you a story that I've never told before. It's actually the first time I've had the privilege to tell it, and it's a story about people, and it's a story about discovery. Apart from the loves of my life, my husband Petter and my daughter Grace, I have two really great loves, and that is people and discovery. I love to meet new people. I love to learn what drives them. I especially love when they have viewpoints very different from mine. I love to have conversations about that. And I really love the process of discovery. I love finding something new every day, and so a lot of the work we do is outside in the field, because when you're outside in nature, you literally see something new at every moment, as long as you're just open to that. So to start this story, this quote actually was the very first lines of my own dissertation, stolen from Charles Darwin's great work. And this struggle for existence is really one of the fundamental ideas of the biological sciences, and essentially it's this. You've probably seen something like this before, either in textbooks or online. This is the so-called tree of life. This shows the diversity of all the species that exist on this planet today, as well as an extrapolation from what species were there through millennia in the past as well, and where we came from. I'm not so much going to talk today about the past, but I'm going to talk about the present and potentially the future as well, which you see is actually at the very rim, this huge diversity of species that we have on our planet, all these different kinds of microbes and plants, fish in the sea, mammals on land, including us as well. It's tremendous how many species exist on this planet. And in particular, I'm going to talk about one group, which you can't really tell is so much highlighted there, but is actually the most biodiverse group on the planet, and that's the insects. So actually, there are nearly as many insect species that exist on this planet today as all other species of organisms put together, which means if you add all the different species of microbes and fungi and plants and invertebrates like crabs and vertebrates like mammals and lizards and birds, they just about add up to the number of insects, which is astonishing. How many insects there actually are, they estimate upwards of 3 to maybe even 7 or 10 million. It's difficult to know because so many of them we don't see. It takes people like you and I to go outside in the world and identify them as new species. We often think of insects as pests that bring disease or destroy our crops, but of course they also serve incredibly important ecosystem services on this planet. They pollinate many of our crops, many of our wild species of plants as well, and they also get rid of a lot of the waste on this planet, like these termite mounds do. They've survived mass extinctions, many of them. They're nearly 500 million years old, the insects are. And we really couldn't survive on this planet without them. Today's story is actually about this little insect. It seems very humble. It's a tiny little fly. And the really interesting thing about this insect is that it didn't exist 300 years ago. It's completely new. And this is the story of how it came to be and what scientists are studying about them. And to tell this story, I actually have to talk about a lot of people that stretch back nearly 200 years in the past who've been trying to understand this process of how species come to be. And I'm going to start in 1830 and go all the way up to the present day, and I'll talk about some of the key players. I'll be skipping a lot of people. Of course, there's many more people involved in this story than I mentioned, but I want to highlight a few of them because they are some of the key players in the story. And like any good story, we can start by saying, once upon a time, two young men bonded over a fondness for beetles. And the reason we know this is they became correspondents much later in life. And here's a particular letter that was written that I can read out loud to you. More than 30 years ago, I was introduced to you in your rooms at Christ College by A.W. Griesbach and had the pleasure of seeing your noble collection of British Coleoptera. Some years afterwards, I became a fellow at Trinity and finally gave up my fellowship rather than going into orders and came to this country. For the last 5 or 6 years, I have been paying considerable attention to the insect fauna of the US, some of the fruits of which you will see in the enclosed pamphlets. Allow me to take this opportunity of thanking you for the publication of your Origin of Species, which I read 3 years ago by the advice of a botanical friend. Though I had strong prejudice against what I supposed then to be your ideas, the first perusal staggered me, the second convinced me, and the oftener I read it, the more convinced I am of the general soundness of your theory. So you may have guessed, those of you who have studied some biology, of who that person was that was writing it. So of course, this is the famous Charles Darwin, who wrote The Origin of Species, which was the first accounting of evolutionary theory that we have. And to summarize really what he was talking about, I'll take a quote from this book that he wrote, Origin of Species. There is frequently recurring struggle for existence. It follows that any being, under the complex and sometimes varying conditions of life, will have a better chance of surviving and thus be naturally selected. From the strong principle of inheritance, any selected variety will tend to propagate its new and modified form. And he also says, a large amount of variability will evidently be favorable for the work of natural selection. That pretty much sums up his ideas in this book. Essentially what he's saying is that no two animals are exactly the same. And because no two animals are exactly the same, they will have different traits. Some of those traits will be more advantageous for certain environments and certain ecological conditions than others. And those organisms will have a better chance of survival and therefore will be most likely to mate. And those traits will then be more likely to be passed on to the next generation, and so on and so forth. And this is essentially the idea of Darwin's theory that this writer was so interested in. So now let's go back to this. And this is actually something that we can see happening right in this very day here in India. If any of you are farmers out there or know anybody who is a farmer, you might recognize this little beast. This is the diamondback moth. This is a huge pest here in India as well as across the world. And it's particularly a problem because it's been getting pesticide resistance. And this works very simply like this. You spray pesticides on the field, you kill most of the insects. The only insects that you leave behind are the ones that can't be killed by the pesticide or are in some ways more resistant to it. So those are the insects that mate and then produce more insects, and then the next generation is more and more resistant until you have a population of insects that you can't kill with your pesticide. And the diamondback moth has become resistant to a number of pesticides, including BT, which you may recognize because it's one of the crops, it's a GMO target for BT cotton and BT brinjal and BT corn and lots of different things. And this particular pest, which is a big pest on cabbages and broccoli and mustard and brassicaceous plants, you can't kill with this. This is Darwin. This is essentially what Darwin was talking about, that these insects are being selected. It's as simple as that, okay? That's the idea. So to go back to this letter, so I talked about who the letter was written to, but who is this guy who wanted to avoid becoming a priest, so he escaped to America? Well, his name was Benjamin Walsh, and you maybe had not heard of him. So he was the first state entomologist of Illinois. He went from Cambridge, where he studied with Charles and others, and he came to the United States to study the insects of the New World. And he is the next character in our story. And his story starts in the 1860s. So this is about the time where he started writing Darwin and saying, you know, I really liked your book. And during that same period, he was studying a number of different species. He was a naturalist. So for those of you who don't know what that means, a naturalist doesn't mean that you just study nature. It's actually a particular way of studying nature. It's also the inspiration for my own lab. And it's that you go out and you observe organisms and you study them for themselves. You study their natural history, where they live, what they do, what they eat, who they mate with, where they mate, what are the environments that they survive in. That's what a naturalist does. They keep their eyes and their ears and their heart open to the natural world, and they observe it. And that's what he did. And by doing this, he observed that there was a shift of this particular little fly, and the scientific name for this fly is Rhagoletis palmanella, and it shifted from its native host, which is a tree with berries on it, it's called hawthorn trees, to introduced domestic apple. And so he said that this may be a new species. So just to show you the actual words of Benjamin Walsh from 1867, the apple maggot is a Native American species and breeds naturally on our wild haws, meaning hawthorns, and crabs, meaning crab apples. But within the last few years has been noticed to attack the cultivated apple of Massachusetts, Connecticut, New York, and probably Vermont also. And what is very remarkable, although the same species exists to my personal knowledge in Illinois, for I bred it myself there many years ago from haws, or thorn apples as they're sometimes called, yet it has not as yet been ascertained to attack cultivated fruit anywhere in the West. It would seem as if this, in many other cases, is only a local race of the species that has acquired the habit of attacking tame and imported instead of wild species, and that this race transmits to its descendants by the law of inheritance the peculiar habits which it has itself incidentally acquired. So, basically what he's saying is that we have a species of fruit that's just been introduced, in this case apple, and that these, yes? Yeah, sorry. What? Oh, it's not, my ears are too small. I'm sorry. You just come run up in me and tell me to fix my ears. Okay. All right. Better? Okay. Thank you. I hope you can hear me better now. So what he was saying is that there's this new group of apple flies that didn't exist before because these apples didn't exist. And instead of just eating both fruits, for some reason they acquired this habit of only liking apples. So now we have a race that only likes apples and a race that only likes hawthorns. Now, where did this happen? Well, it happened, among other places, in New York. The Hudson Valley is in New York. That red dot up there is actually where I was born. I was born in very northern New York State in a farming region. And this is where the apples were in pre-colonial times. If you don't see anything, that's because there was nothing there. There were no apples, okay? Apples are introduced to the New World by colonists. They did not exist in North America before this time. And by 1875, there were literally millions of apple trees. So about the time that Benjamin Walsh was sitting outside and observing this change, there was tons of apple trees around. And what is very interesting is that instead of these flies actually just liking two things, you know, like I can eat idli or dosa for breakfast, you actually just choose one of them. So we have this group of flies that only likes apples and this group of flies that only likes hawthorns, and they're somehow different from each other. And this is the idea that he called phytophagic races, which means plant-eating races of flies, okay? And Charles Darwin was so admired this idea that he himself changed his Origin of Species in its 4th edition to incorporate Benjamin Walsh's ideas. They continued a correspondence for a number of years. It's lovely, lovely letters that you can read in the Darwin Correspondence Project at Cambridge. And among them is this note that he changed his work. And he says, B.D. Walsh, a distinguished entomologist in the United States, has lately called attention to some cases analogous with those of local forms of geographical races, yet very different from them. These cases have been fully described under the terms phytophagic varieties and phytophagic species. Most vegetable-feeding insects live on one kind of plant or on a group of plants, and some feed indiscriminately on many wildly different kinds. Yet this induces no change in them. But Mr. Walsh, however, has observed in other cases in which the larvae or the mature insect, or both, are thus affected by slight though constant differences in color or size or nature of their secretions, and then become two different species. So this is the idea behind it. However, while they both talked about this idea, neither Darwin nor Walsh actually got into how this could happen. They talked about, yes, different variation, there being a lot of different types of traits in a population, and some are better adapted than others, but they don't actually talk about how species happen. This is just about how species can change over time, but it doesn't talk about the process of speciation itself. And for that, we actually have to skip ahead to 1942, and we have to go to New York City, where there was a man who happened to be working at the Museum of Natural History in New York City, and he is very famous because he's considered the father of modern synthesis of evolution. And his name was Ernst Mayr. And Ernst Mayr wrote a book in 1942 called Systematics and the Origin of Species from the Viewpoint of a Zoologist. He was particularly interested in birds. He was an ornithologist. And he, through looking at different forms and structures of animals out there, came up with the idea that the crucial process of speciation, which is the birth of a species, is not selection, but actually isolation. So it's that the species have to be separated from each other somehow. And he came up with this idea. So I'm going to take you to Kashmir. This is one of our field sites. It's very beautiful, so that's why I want to show the picture. And this is a hypothetical situation, but it's what Ernst Mayr based his ideas on. So you have these two butterflies, okay? And let's imagine these butterflies are separated on these two mountains. And they're separated by this river, the Zojila Valley. And they can't cross it for some reason. Maybe they can't get across the river, okay? So they're stuck on either side. And that little village there on the right maybe produces a lot of smoke, and so over time, one group of these butterflies starts to get dark, which we actually have seen happen. So this is a very famous case in the peppered moth, that it turned colors because of the industrialization and the air pollution. But because those butterflies on the right can't get across that valley, they no longer can mate and reproduce with the butterflies on the left. So that color change only stays with them. It never goes to the other side. And because they're physically separated in space, they can't mate with each other. Over time, they would accumulate enough traits that they might become different species. And this is called geographic isolation, so separation in space. And this is how Ernst Mayr considered that new species could come to be. So of course, selection is important. These butterflies are under different selection pressures, one under the pressure of the smoke and one not. But it's actually the fact that they're separated in space, this isolation, that's actually the key factor that makes them new species. But if we go back to our flies now, this fly story that I was telling you about, these flies are not separated in space. They're sympatric, which means together, okay? They're never isolated from each other. So apple flies and hawthorn flies are always within flying distance to each other. And in fact, I can tell you, because of where I grew up, these apple trees and hawthorn trees are literally standing right next to each other in fields in the United States. So there's no separation. So Ernst Mayr says, basically, that's not going to happen. This is not actually going to become two species. These are just going to be some flies that happen to choose both things. That's the idea at this point. So the next part of our story, we have to skip ahead to the 1960s. To go to Harvard. So Ernst Mayr eventually took a position at Harvard University. He again wrote many, many other works about his idea about isolation, which are fundamental to our understanding of evolutionary theory. And at one point in 1960, he got a new student. The student didn't look like this at the time. I can't find a young picture of this guy, so I'm sorry, this is the picture I have of him. But he's very integral to this story. His name is Guy Bush, and you might see behind him in the picture who's sitting there behind him. It's Benjamin Walsh, right? So he was actually a student at Harvard at the time, and he took this class of Ernst Mayr where Ernst Mayr talked about this whole process of isolation, this modern synthesis of evolution, and he was really just fell in love with it. He was like, this is amazing, this is such an exciting thing to study, and he also was very interested in fruit flies. And so he knew about these apple flies. So he set out actually to show that this was not happening, that Benjamin Walsh was wrong, and that this were not actually becoming new species. So, like any good naturalist, he went outside, he watched these flies, he looked at where they lived, he looked at the flies on the hawthorns, he looked at the flies on the apples, he looked at what they were doing, where they were flying, who they were mating with, and all of this. And he observed something very interesting. So the flies actually grow up on the fruit. So this is an apple, as you can see, and the larvae, actually, the maggots, actually eat the apple. So this is the proverbial worm in the apple. So they're a huge problem, actually, in the US. They're one of the biggest pests in the United States on apples because of this, because you don't want one of them in your apple, or more worse, a half of one in your apple. Okay, so they actually mate on the fruit, okay? And they lay their eggs in the fruit, and then the fruit falls to the ground, and that's where the larvae emerge as flies the next year. So they stay overwinter underneath the soil where they're not frozen. The key thing that he noticed was actually that they actually mate on the fruit itself. And then he got the idea, well, actually, this may be possible to be isolated because, for example, if you're an apple fly and you really like apples and you only go to apples, well, then you're only going to mate with other flies that only like apples because you mate on the fruit. If you're a hawthorn fly, you go to hawthorns and you only find flies that like hawthorns because an apple fly would never be on a hawthorn because it doesn't like it. And he came up with this idea that this actually could be possibly separating them, and he called this ecological speciation. But it was very controversial at the time because it's not really clear how this can happen, right? How can these flies actually be so separated when they're so close together? And particularly when they're only 300 years old. This is really short period of time. I mean, most evolutionary stories we hear about are talking about millions or billions of years. This is 300, right? This is like in our time, in our recorded time. And so he was very nervous saying anything to Ernst Mayr, because Ernst Mayr even went so far as to actually make a statement about this in the 1962 book, right in the middle of Guy Bush's PhD. And he said, one would think that it would no longer be necessary to devote so much time to this topic. Sympatric speciation is like the Lernaean hybrid, which grew 2 heads whenever one of its old heads cut off. And here you have, can you imagine if your professor is writing this in one of his acclaimed books and you're writing the exact opposite thing in your dissertation? How scared he must have been. He was completely intimidated. So he wrote and he rewrote and he fixed and he refixed his dissertation, very nervous about submitting this to his committee member, Ernst Mayr. And in fact, he took so long that Ernst Mayr was about to go on vacation when he finished his dissertation and said, "You can only hand in your dissertation to me if my porch light is on." And here's what happened. He told me, this is Guy Bush's words, he told me that if his porch light was out, it would be too late for him to read my thesis, and indeed, the porch was dark. So Ernst Mayr never read his dissertation on these flies. He never read his ideas about sympatric speciation, never read about the mating on the fruit. He never actually accepted his thesis. So actually what Guy Bush had to do was go and find another person at the very 11th hour of his dissertation work to sign off as in his thesis to allow him to get his PhD. So that's what happened with that story. But Guy Bush persisted, and he still believed in this idea. He still believed that these flies could, in fact, be becoming different species, that actually might be something there, even if they weren't separated in space. So he went out and he became a professor himself, and he got his own students, and he started trying to figure out how this could happen. What is the mechanism that is keeping these flies on these different fruits? And for that, we skip to 1975, to this wonderful man right here. And you might see on his shirt, there's a little apple fly on his shirt. His name is Ron Prokopi, and he was a crazy and wonderful man. He's a behavioral ecologist, and he came up with this Prokopi Bioexperimental Farm, which is literally, he parked a trailer in the middle of an apple orchard so he could watch these flies day and night and observe exactly what they were doing. And he did these wonderful experiments, like hanging wooden blocks and wooden spheres and wooden squares in a tree and covering them with sticky tack so that flies would get stuck to them, just to see what shape flies like. He made giant cardboard cutouts of trees. He made them tall or round or blue or purple or green, just to see what colors and shapes that these flies liked for their trees. Very simple experiments, needing just glue and wood and maybe a little ingenuity, but so elegant, and revealed so much about what it is about these flies that they like about these fruits and these trees. Because like I said, they mate on the fruit. It's actually the fruit they like, not each other. They only get to each other once they get to the fruit, okay? So it's really, the fruit becomes their whole world, and he studied that. And one of the most important things that he found, at least for our story today, he found out many things, but for our story, the important thing is that they actually like the smell of their fruit. So, many insects, when they're flying around, they can't see from very long distances away. They have really little eyes, right? So their acuity is going to be limited from a distance. They're very fast vision, but it's not very accurate from a distance. So we also have limited vision from very far distance, so we might use something like sound, right? We might hear calling or yelling from a very long distance when we can't see something. So insects don't have ears like us. They have a different type of hearing. They detect vibrations. So for them, from a distance, they use their sense of smell. So it's actually the smell of the fruit that actually guides these flies to their different hosts. And he found, Ron Prokopi in the 70s, that apple flies really like the smell of apples, and hawthorn flies really like the smell of hawthorns, and they really hate the smell of each other's fruits. And that's really amazing because, again, these flies didn't exist 300 years ago. The apple fly is completely new. So how does that happen? Well, now we need to bring in chemistry. So now, how is it that these fruits have different smells from each other that these flies don't like and that they go to each other's fruits? We will skip ahead to 1990, where we'll go to Geneva, New York, which is actually an experimental station that's in collaboration with Cornell University, to this man. His name is Wendell Roelofs. Wendell Roelofs is considered the father of chemical ecology, along with a man named Thomas Eisner and Jerry Meinwald, who all were professors together at Cornell University. And Wendell was famous for identifying volatiles that were important for insects. He particularly worked on pheromones, but he did a lot of other work in other systems as well. And one of the systems that the experimental station was working on was apple flies, because New York is a huge apple growing region. There's a lot of apples growing there, and so these flies were a huge problem. So basically, this experimental station, which was done for farmers, was just trying to kill them, trying to get rid of them in some way or another. And so Guy Bush came to him and said, "Wendell, could you help me identify what are the volatiles? What are the smells coming from these fruits for these different hawthorn and apple flies?" So that's what he did. So he used a variety of techniques by collecting the odors or the head space around these fruits, and he identified the different chemicals. And this actually is a synthesis of about 30 years of work in this area, but the cool thing about it is this. So apple volatiles are different from hawthorn. They don't smell the same, for sure. However, if you look among the different varieties of hawthorns, and that's something I haven't told you before, there's actually different kinds of hawthorns out there. And if you look at the different varieties of hawthorns, if you take a little bit of this, a little bit of that, a little bit of that from the hawthorns, you can make an apple. So that means that the apple flies were, although smelling something different than the hawthorns, it was not completely new to them. The apple was some sort of mishmash of a bunch of things they already were able to smell. And this is important because this means that they didn't have to completely change everything about their lifestyle. They actually could take stuff they already had the ability to smell, and just they'd have to change how they were detecting it and how they were responding to it. So that means an apple, while new, was already there in their abilities all along. And that's what is shown here. So you can see the green from the apple flies is there in the hawthorn flies, but it's not exactly there. There still is a change. But again, this still doesn't answer this question. How did we go from a fly that loves hawthorns and hates apples to a fly that hates hawthorns and loves apples? how do we go from this kind of a switch? So now we have to go to this century, to 2005, to this young woman who you may recognize. So I joined Wendell's lab in 2000. I was a chemist. My bachelor's degree is in physical chemistry, to be precise, but I was really interested in how chemicals could be used by organisms in their lives. And so this story fascinated me. I was really interested in this story because I was wondering, how is it possible that these volatiles are being used by these different flies? And how is it that you have apple flies and hawthorn flies and they're different from each other? So to study that, I started doing physiological work, neurophysiology, on the antenna of the flies. So insects have a nose, just like we do, but it doesn't look like this. It's antenna, right? The sense of smell for insects is actually their antenna, which is colored green in this image. Of course, it's not really green. It's just so it shows up on this slide. And you might notice on the surface of this antenna, it looks a bit fuzzy. And that's because there's actually a lot of little tiny hairs on the surface of the antenna of insects. Each of those hairs contains neurons. And each one of those neurons binds to a particular type of smell. Just like we do, we have a bunch of neurons in our nose that can smell coffee, or can smell tea, or can smell bread, or can smell all sorts of nice and bad things. And when we smell something, the molecules from that object literally go into our nose and bind to our neurons, which is why it's hard to smell when you have a cold, because your mucous membrane thickens, and the chemicals literally can't make the trip all the way to those neurons. That's why it's hard to smell. It's not that your nose has gone bad. So these guys have their nose on the outside of their body. So instead of having to breathe in, it's just passive, and the chemicals just go to their antenna. And so I decided I was going to look at these antennae and look at these apple and hawthorn flies, and I was convinced I was going to find the key to this 150-year-old story. I was going to answer it. I was going to find the difference between these different flies. And I did not, okay? So there is no difference between these flies' antennae at all. In fact, this graph, which is a little bit complicated, but it says something very simple. I looked at flies that were reared in the lab. I looked at flies that had been collected from the wild. I looked at hawthorn flies. I looked at dogwood flies, which I haven't even told you about, are just another species. I looked at blueberry flies, which are another species, and they're all exactly the same on their antenna. They have the same neurons. They can smell the same things. They can smell them in the same sensitivities. I could see absolutely no difference. In fact, if I didn't know where those flies came from, I wouldn't be able to tell the difference between them. They seemed exactly the same. So unfortunately, I had completely negative results for my dissertation, but I still managed to get a PhD anyways. So the story was not finished. And I went on and I worked at Max Planck, and I spent a number of years learning lots of different techniques in the hopes that someday I could come back to this story. But in order to come back to this story, I had to come back to it once I got here to Bangalore. And so this story continues just until this year. And this particular young woman is named Cheyenne Tate. Cheyenne Tate is a joint graduate student between myself and a man named Jeff Fader, whose advisor happens to be Guy Bush, who I told you about before. So this small story comes back to the same types of people, and she came to my lab to look not only at the surface of these flies, but actually inside of them, in their brain, because there must be a difference somewhere. The way that we have different behaviors is because of the brain. It's the brain that leads to behavior. So if these flies actually are different from each other, it has to be occurring somewhere in their nervous system. And so we decided to look at the brain, and there's 3 basic places where there could be a difference. You can have a difference at the level of the sensory receptors, which I already told you was not really happening. The sensory neurons could go to different places in the brain, or they could be processed differently. Maybe what these flies think about these volatiles is different. So that's what we set out to do. So first we had to look at the brain. So this is what the brain of an insect looks like. So this is a staining of an insect brain. This is actually an apple fly brain. And the different colors of the stain are actually staining different regions. The red stains all neurons in the brain, and the green is staining particular neurotransmitter, and it just allows us to get a contrast. So we do this by dissecting the brain out, and then using this dye, and then doing something called confocal microscopy, which allows us to see a very detailed level in the brain. And you can see there's all these little structures in the brain, right? You can see this area that has a lot of balls inside of it. You can see this area that something looks like a flower maybe. And these structures all govern different aspects of the fly's life. They're for taste, and for smell, and for sight, and for memory, and for movement, just like our brains. Because an insect brain fundamentally is not that different from ours, except for being much, much simpler. They only have 100,000 neurons, we have 80 billion, but they still have places where they process vision, and process olfaction, and process movement, same as us, just at a much simpler scale. So we reconstructed the brain, basically using the same type of technology that you do for CGI. So basically this is just very early days Bahubali, okay? So what you have here is the brain of these apple flies, and it looks a bit like a Batman symbol, okay? And if you take the wings of the Batman, those outer portions in taupe, those are actually the optic lobes. So that's the region of the brain that processes the sight from the fly's eyes. Those two balls in the middle, the red balls that you see, those are called the antennal lobes. And you probably have guessed, they process what comes from the antenna. So they process the smell. The green squiggly lines are called the mushroom bodies. It's a weird name, but that's because they were first found in bees, and in bees they look really exactly like mushrooms. In here they look just kind of like weird things, and they process learning and memory for the flies. And the donut and the teal and the mustache in the back, those are the sensor-motor centers of the brain. So these are the areas of the brain that take the information from the sensory system, convert it into movement, and allow the fly to then make decisions like flying. So these are the areas that we concentrated on. We thought, okay, well, it's either where they smell, because I told you smell's important, what they remember and learn about the smell, or how they move for the smell. And so we decided to look at one of these regions first. So we started, of course, with the first processing region, which is actually the antennal lobe. Now, this is what it looks like, okay? So again, it's a CGI Bahubali version of the brain. But you can see right here, all these little balls inside the brain are all different regions of the antennal lobe. And each one of those balls takes information from a particular type of neuron. So if you have a neuron that responds to this chemical, it will go to one of those balls. A neuron responding to a different chemical will go to another ball. So it's like a map. It's essentially a sensory map of the antenna. So by looking at how many balls there are, we know how many chemicals there are that are detected by that organism. We have the same thing. We actually have about 300 receptors, and we have an olfactory bulb that looks exactly like this. It's just obviously a little bit more complicated, but it's the same thing. So do fish, so do most organisms. This is a conserved way of processing smell throughout many of the animal kingdom. And so we thought, well, of course, there's going to be at least one or two regions that are going to respond to those apple volatiles, and one or two regions that's going to respond to those hawthorn volatiles. But this tells us nothing, because the apple flies and the hawthorn flies look exactly the same at this level. There's no difference in the way their brain looks, but it must be some difference in how their brain works. So we have to do functional brain experimentation. So for that, we turn to this, okay? Not exactly this, because we can't do this on flies. As you can imagine, they're very tiny. They don't fit in that machine. This is called a magnetic resonance imaging instrument. This is actually for functional magnetic resonance imaging. If you haven't seen this before, you can just look it up online. You can look for lots of videos for this. This is used in humans, particularly, but it can also be used in mammals, to identify where regions of activity are in the brain. So you put a human into that scanner, okay, and then you can talk to the human. You can talk to the person, I should say. You can play it sounds, you can play it sad movies, and you can see which regions of its brain light up. And that tells you where it is thinking at that moment in time. So basically, we wanted to do the same thing with our flies. We wanted to ask them questions by presenting them different odors and seeing where in their brain they were thinking about those smells. But like I said, you can't stick a fly in here. Okay, it's too tiny. Their whole brain is actually less than 1 pixel of an fMRI machine. So we have to use a technique called calcium imaging. Okay, so calcium imaging is a technique where you can also see the activity of the brain. Calcium is a very, very important chemical for the nervous system. It's actually used in the brain, and it's exchanged whenever you have a synapse. A synapse is where neurons come together. So whenever you have an active synapse, meaning you're thinking, you will have an exchange in calcium when there's a chemical synapse there, and that will then be registered by our technique, because we have put a small dye in the brain that changes color whenever there's a change in calcium. So whenever there's a change in calcium, we know where the brain is thinking. And finally, after 150 years, we finally see a difference, okay, with this brain. So this is actually these, do you see these balls over here? They're not so clear as they were in my 3D reconstruction, but those are the antennal lobe. And we presented living flies. Of course, they had to be alive to think, although some people don't think when they are alive, but in this case. So we had the hawthorn flies smelling hawthorn odor and smelling apple odor, and the apple flies smelling hawthorn odor and apple odor. Okay, so we had two different odors, types, and two different flies. And it's a little hard to see. I know it looks a little bit like a Rorschach test, but I hope you can see that the top left and the bottom right have a very big ball of activity, and the top right and the bottom left have much smaller balls of activity. That's because they've switched where they're processing information in the brain. So where the apple flies smell apples is where the hawthorn flies smell hawthorns. Where the hawthorn flies smell apples is where the apple flies smell hawthorns. So they've just switched the region of the brain where they think. Okay? Nothing else has changed. Nothing has changed in the structure, nothing has changed in morphology, nothing has changed in the sensitivity. It's just that where the information gets processed gets switched. In other words, if you change what you're processing at the go and the stop, you will suddenly hate what you used to love and love what you used to hate. So the apple flies now process information where the hawthorn flies used to hate it, and vice versa. So just by a very, very simple switch, you can actually change your mind and change your species, just like that, by a very, very simple switch in the neurophysiology of the brain. So this system has gone from being a model for evolution and a model for speciation to now being a model for neuroscience. Because if you can change your mind like that, that becomes extremely interesting to neuroscientists, because now we can look at what are the properties of the neurons, what is the properties of the pathways that can actually change and cause this to happen. So this system is now evolving now into not only a system for evolution, but a system for other fields of science as well. And there's lots of outstanding questions that we have. For example, we don't know the genetic basis for this switch. The genome is just being sequenced as we speak, and now we're going to be able to look at it, and now we know what to look for. We can look for changes in the nervous system, and that can give us clues onto how these flies switched. We don't know how these flies came about. Were they always there? Was there always a population of flies that just happened to hate hawthorns, even though they were supposed to be on hawthorns, and when apple came, they went to it? Or was there just a broad variation, and there was flies that didn't care? We don't know. We're trying to do these experiments as well. And at least for our lab, one of the questions that we're most interested in is actually, how do these flies go to this fruit? How is it that they go to these fruit? What is it that's important to them? Is it the way the tree looks? Is it the way the tree smells? Is it a combination of both? We know that smell is important. Is it always important? What if you smell an apple tree and you get to it and it's a hawthorn tree? What do you do? This is very hard to do in real life, as you can imagine. It's very hard to make a tree that suddenly changes smell, or a tree that suddenly produces different fruit. Even with all the GMO technology we have today, we haven't produced a tree that can spontaneously shift into something else. So for that, we use virtual reality. So this is a technology that we've developed in our lab to allow us to be able to present these flies with choices in a virtual world. So we don't present them with a real tree, we present them with a virtual tree, and then we watch what they do. So the fly actually makes a decision, and we just watch it, just like we would do in the real world, but in this case, they're in a virtual one. Maybe you've all used virtual reality goggles. There's Smash here in Bangalore you can go to and try these on, and you can drive a motorcycle and all of these different things. We do the same thing, but for an insect, okay? This is how it works. This is the arena. This was built by Pavan Kaushik, who actually is a Bangalore born and bred, and he built this virtual reality himself, along with a few others in the lab. This is what it looks like. The first most important part is the fly. The fly is held in place. It's alive, of course. It can fly, of course, but it can't fly away because then it flies out of the world, okay? So it has to stay in the world. We have a camera to film the behavior of the fly as it flies around. And we have a panoramic display. This is really important because insects have huge eyes. Many insects can actually see in the back of their head, and that means our virtual reality can't be goggles. Well, I suppose we could, but I don't want to make goggles for a fly, okay? So we instead made an arena that goes in 360 degrees around them. And we, of course, also have wind and odor. That's very important because, as I told you, the smell of the fruit is really important to them. And the way the smell gets to them is through the wind. So here's what it looks like. This is what it looks like, the cockpit of a virtual reality arena. This is what we see on the computer screen. Here's the fly here. This is a still image, so it's not moving yet, but it will. And we have the world there. And it looks a little distorted, and that's because it's actually wrapped around in 360 degrees. So like a world map, it looks a little bit strange when you unwrap it. And how we know what the fly wants to do, how do we know where the fly wants to go in this virtual world, is by how it moves its wings. If it wants to move left, it will beat its wing really hard on its right. If it wants to move right, it will beat its wing really hard on its left, just like it does in the real world, okay? And then we move the world in response to that. So we don't move the world until the fly tells us to do so. And by that way, we know what the fly wants to do, whether it wants to go this way or that way, whether it wants to go to the tree or away from the tree, etc. On the right there, you will see the trajectory that the fly will take. The lower dot is the starting position of the fly, okay? And it can actually go in any direction, and this is the meters, literally, that it would be going if this were a real world. It's of course not going anywhere, it's in place. But if this were a real world, this is where it would be flying. So here's what it looks like. So you can see the fly is now making the decision to go to this tree. Okay, and it's flying in and around the tree, and it's maybe looking for apples. We don't know what it's doing exactly, we just know how it's responding to these stimuli. And you'll see in the next example, we start over, and you can see we present it, and it goes straight for this apple tree. And if you watch the fly really closely, you'll see that when it gets very close to the tree, close to the leaves or close to the fruit, it throws out its legs in front of it. And in the real world, a fly does this for two reasons. Either it's about to crash into something, or it wants to land on something. And this is how we really know that these flies actually see this virtual world as if it were real objects, because they're actually trying to land on the apples. But there's no apples there, right? This is virtual reality. So we're using this to try to understand how these different flies, these apple and hawthorn flies, actually make decisions, try to understand their brains a little bit better from the standpoint of the fly itself. So we're trying to take this idea of naturalist inspired and put it into this artificial context and present them with all of these different choices and see through their eyes and through their antenna exactly the type of decisions that they make. And this allows us to have a whole new field opening up on decision making. Decision-making is a really important field for a lot of people. Driverless cars, they're very interested in this technology because they have to decide whether it's a human being or a lamppost or a stop sign or not, right? It's very interesting for search algorithms. Search algorithms are inherently tied to the ideas behind decision-making. It's very important for understanding neurological diseases. So then now we're going from these flies from an evolutionary perspective into even the perspective of computer science. So this story really started a long time ago in a dorm room in Cambridge and went through a whole lot of different people who are doing a lot of different techniques. We have everything from the HMS Beagle that Darwin took around the world to learn about all these different species on this planet and get the idea behind his Origin of Species, to the entomologist in New York who studied these flies and first found them to be a potentially new species, to the systematist at the Museum of Natural History who realized that isolation is very important, to population genetics from Guy Bush and the behavioral ecology of Ron Prokopi, all the way up the chemistry from Runda Roelofs, and the neuroscience that we're doing today. So when you read a science story, I hope you realize that it didn't start with that lab that you're reading about. It started 100, 200, 500, sometimes thousands of years before that. And when we say that we stand on the shoulders of giants, it's not just an expression, it's really true. Every bit of scientific information, every new discovery that we make today is built upon all of the knowledge that is built up before us. And it's very rare that we have the opportunity to bring out this history. And so it's been a real privilege for me to tell you this story today and to honor all of these amazing scientists that have come before me to help me to learn this exciting thing about these flies. Because even our virtual reality would never have existed without Charles Darwin. So I'll just end with this image of this humble fly and all of the work that's gone into this fly, and also thank a few people. One is Charlie Lin, who I didn't mention, who is a fabulous scientist at Cornell and one of my mentors, and has done most of the behavioral work on the flies in recent times. Jeff Fader, who is a student of Guy Bush, who I collaborate with, and who we share a student, Cheyenne, who did all the imaging work I showed you. And of course, this amazing group of young scientists I get to work with every day, who inspire me to reach new discoveries and whose shoulders I stand on. Thank you so much.
Thank you, Shannon, for this fascinating story. It's a really wonderful talk. Are you willing to take some questions?
Yes, of course.
Yeah, so
I hope we
please
can answer them.
ask a lot of questions and let's hear more of our fascinating journey in science.
Hello, thank you, ma'am, for the wonderful lecture. Does this work prove that, the speciation actually opening up of new niches and alternate use of existing organs? Does it, give an alternate theory for speciation? That's question number one. And number two, you were insisting upon the olfactory centers of the fruit fly's brain. Sorry, I got hooked to fruit fly. The apple fly.
It's a fruit fly. It is called a fruit fly. It's called a true fruit fly, actually.
Okay. Drosophila. Okay, I think there should be some correlation with the motor areas and all optical areas because the fruits themselves look different and the kind of
Yes.
usage it has to do. And third question is, initially, when you started the lecture, I was thinking that the males kind of mark their territory on an apple and females tries to go towards it, probably use of some pheromones and so on and so forth. Is that something that you have observed or what's the deal about it? Thank you.
Try to do it backwards, okay? So I'll answer your last question first. There's no evidence in this particular group that they use pheromones from a distance. It seems very clear that they use the smell of the fruit. So for example, if we take a fake apple, right, and we give it the smell of the apples that we've identified, the flies will go to it just as they would to a real apple, whether or not an insect is there. So it doesn't seem to be the long-distance pheromone. You are right, however, that the males come first. They actually have an interesting behavior that's been found in several animals called lecking. Okay, lecking behavior is a really interesting behavior where the males actually have displays. So in this case, their fruit is their display. So they find a very pretty fruit and they sit on it all day. I have sat in 35-degree heat plus in Pennsylvania watching this male who never could get a lady all day. 8 hours a day he sat. And at 4:30 p.m., he'd been there since 7 a.m., he finally got a lady to come, okay? So they're very patient, these flies. And the males sit on the fruit, and then finally a female comes to the fruit, and then the male comes and gets her, grabs her, and they mate, and then they have babies. But there's no evidence for pheromones, okay? The olfaction part. So absolutely, there is other things there. That's what our virtual reality, and I'm glad you asked that question because I didn't go into great detail about that. There is actually a lot of differences in these fruits besides their smell. The trees are different shapes. Apple trees are kind of like mango trees. They're quite round and short trees. Hawthorn trees are big, tall trees, okay? They're very, very different shaped trees. The fruits are different sizes. Hawthorn berries are really small. They're even smaller than a blueberry in size, whereas you know what an apple looks like, right? So it's a much different size fruit, different colored fruit even. And so all of these play a role. And Ron Prokopi, who I mentioned, he'd done all these experiments and he'd shown these differences are at play. So absolutely, this may not be the only difference we're seeing. It's definitely one of the key differences, and it's the first time we've seen a real difference after, all of this work that's been going on. But there could be more differences, and that's actually something our lab is working on. And the first question I already forgot. What was the first one?
Okay, the speciation is opening up of new niches and alternate use of organs. So is that a powerful mover in speciation?
The ultimate use of what?
Of existing organs, because you were telling that the olfactory sensors were already in place. It's just the mix match that
Look,
create
yeah.
niches.
So look, if you're going to go to a new region, okay, so for example, if you're going to choose another fruit, you don't just spontaneously gain the ability to detect that fruit. That's crazy, right? You have to have some standing ability to detect something from that fruit as being worthwhile to you. So that's kind of an obvious thing, right? So if you have an invasive species, which is a huge problem here, and actually this story is about an invasive species, in this case the invasive species with apples, but invasive species of plants and animals are very common here in this country, and one of the things that our lab studies. Most of our work is on Indian systems. This is the only non-Indian system we work on. And in those systems, right, the invasive species cannot become a pest or a problem unless it can already detect the things that are there, right? It's not going to start attacking this plant or this animal or whatever it is unless it already has some ability to recognize it as an object of its interest. So, yeah, so it has to be there.
That was fascinating. So 2 points that here.
Oh,
Here,
hello,
yeah.
okay.
Yeah. So 2 points. I think the first point that still kind of intrigues me is how the combination of existing hawthorns somehow match exactly an apple, which well, from my mathematical training, I'd call it a linear combination. But it's interesting that that an apple is completely represented by a bunch of different hawthorn things. That just brings me to a question of whether there exist other fruits that can be completely mimicked by some other fruit. So there's a sort of a basis of fruits that represent all possible smells, and then there are some other fruits which just happen to be combinations.
Yeah, I'm glad you asked that question, okay? So these apples don't smell like hawthorns. Apples produce hundreds of chemicals, okay? And actually the smell of that we think is an apple obviously smells nothing like what an apple fly thinks. Because if I give you that blend, you can come to my lab. I invite you all to come to my lab at NCBS. I will give you the apple blend, and you will say, that doesn't smell like apples. Because it's not for us. That blend is made for these flies, okay? So what these flies detect in these apples is found also in their hawthorn fruits, but it doesn't mean they smell the same, okay? It's not mimicking, there's no correlation. There's tons and tons of volatiles in apples that don't exist in hawthorns. What I'm saying is that the things that they pay attention to are similar. Just like us, right? There's lots of chemicals out there that we can't smell. We can't smell cockroach pheromones, okay? Which is a good thing for us. But that's because we don't have the ability to detect them. We don't have the sensors to detect them. That's what cockroaches have, and good thing for them. So because we don't have the same sensory system, that's what we have. We are humans. We have the sensory system that helps us to survive in the world. Those fruit flies have a sensory system that allows them to survive, and it's not the same. So that's what it is. So don't misunderstand me that they're actually smelling the same. They're not at all. It's what the flies pay attention to that has some correlation.
And just one more point, I was reading this work by V.S. Ramachandran, the neuroscientist, and
Yes.
he was talking about...
The telltale brain,
yeah, so the phantom brain, yeah. And he
event brain.
And he was referring to the phantom limbs where there was rewiring of
Yes.
neurons from parts of the hand to parts of the face. So as in a thought experiment, but there could be ways of rewiring the apple fly to be a hawthorn fly, if that can ever be done.
Theoretically, yes. Yeah, that would be an awesome experiment because, I mean, the phantom limb syndrome that you're talking about, right, actually is a much more complex phenomenon. Actually, it can cause rewiring at multiple levels, both the sensory level or the central. So there's a cortex called the somatosensory cortex in the human brain, and Ramachandran proposed that it's actually the somatosensory cortex that's changing. This is much simpler. These flies have way fewer neurons. We actually think it's actually only one neuron or two neurons that are actually changing, okay? It's only a few neurons that need to change places in order for this love-hate relationship to happen. So potentially, it's possible. One could even imagine there's this new technology out there called CRISPR, which is this really amazing technology where you can introduce genetic changes into an organism. Potentially, we could do that and do this experiment and see if our change works, right? If we could change the fly's brain and it suddenly goes to something new, then we figured it out, right? That's the proof in the pudding that we actually understand this change. I would love to do that. you can come to my lab and figure out how to do CRISPR in these flies if you want, because I don't know how to do it. So, but there's tons of questions that we can pursue with the system.
Hi, thanks for the wonderful talk. Here, yep. So how does the mating preferences co-evolve with the food preferences? Because in order for the speciation to occur, we need this mating preference. So is it something to do with olfaction, which has a similar role in both? Or?
So this is a cool thing I didn't tell you about. So if you take these flies, so you can take them into the lab, right? You can take the fruit into the lab and rear the flies through to adulthood. So if you take them into the lab and you put a hawthorn fly, a male hawthorn fly there, and you give them a female apple fly, they'll mate easily. No problem. There's no difference in their mating, okay? They don't care once they're together. So that's what the dangerous thing is, and this is why the whole idea was controversial, because it's not that their mating is different, it's that where they mate is different. So the only thing keeping these flies separated from each other is the fact that they like different fruit. So the apple flies only go to apples, and the hawthorn flies only go to hawthorns. If you artificially put them together, they'll mate fine.
So what kind of progeny do they produce?
Their progeny doesn't like anything. Truly,
Okay.
they can't find fruit. They don't respond to these host volatiles, which we think now, based upon our neuroscience, we can come up with a neurological reason for that there's a miswiring happening. So that's where we have to go next.
They have a poor selection.
They don't select, they don't select. So the idea, of course, is that if these flies existed in the wild, they'd be at a disadvantage because they're not able to find fruit. So that's the idea. And if you cross those with each other, okay, if you take those, if you keep them in the lab so you can keep them alive, and they'll stay alive and they'll mate, those progeny, you get an F2, which is grandchildren, that's what we say. F is the parents, then F1 is the progeny, and F2, and then F3, and so on. Then you get something called Mendelian inheritance, okay? You get 25% of the flies roughly that like apples, 25% roughly like hawthorns, and 50% that don't like anything. And then you have to go back to a man named Mendel who came up with this idea with pea plants. Right. And that suggests that there's a single gene controlling this. Right. So the saga continues. So.
Ma'am, to your right. This is perhaps a much more basic layman type of question than what you've heard so far. So to me, when you showed the calcium experiment, it looked like you had two possibilities. One is that the area which was initially apple ready would just expand and become more ready to become larger, or what you showed was like a railway track change where it actually went back to the other side. But why do you think that happened?
So we did a series of experiments. So because I didn't want to bore you guys with tons of neuroscience, I didn't show you all the experiments we did. We also did single neuron staining. So what we would do is we would actually take an electrode and insert it into this region of the brain and record neurons, and we recorded neurons that responded to apple or responded to hawthorn, and then we injected a dye into them and traced the pathways, and that's how we know they're switched, because we could see that the pathways themselves are switched. Your idea is completely possible, but it was verified through these other experiments, which I didn't mention actually. So
do you have a theory of why that would happen rather than just maintaining the same pathway and exciting that same?
So why that happened, so why questions are notoriously difficult to answer in science, right? We answer how questions and what questions are pretty straightforward because they're hypothesis testing. Why is a very, very hard question to test. I can say, in all honesty, right now, we don't know, okay? But there's two basic possibilities. One is that in the hawthorn fly, way long ago in antiquity, there was this low-standing population of flies that for some reason hated hawthorns. I propose, this is my own idea, that it might be to help them to find fruit in the event that their hawthorn tree is not available. Now, something I also didn't mention in this story is that hawthorns don't fruit every year. They can actually go 2 or 3 years without fruiting, okay? There's also flies that can survive for 2 or 3 years underground, so that also happens. But if you happen to be a fly that comes out and there's no hawthorns around, what are you gonna do? So having a fly that's willing to accept other fruit actually could be very advantageous. But you don't want too many of those, because then you won't find your mates again, right? Because you're gonna only have those flies that are on the fruit mating. So I don't know if that's the case. That's something that you can test. We can do a lot of behavioral experiments to test this, and this is really where we're going to go now. This research is not even 6 months old, so we just found this out. So there's a huge amount of experimentation that can go. But why questions are always hard to answer. So,
Ma'am, what do you think about human evolution? Can we truly evolve to form a new species, since we live in a truly global world where anyone can mate with anyone else?
well, what do you think? What do you think?
I think we cannot, because mostly, if you see, species evolve because of isolation, or okay, I just know about isolation for now.
Yeah, I mean, so obviously we are all one species. We're all humans. We are all one group of people. We are all the same, and we do travel. Like, look, I live here in India, right? I mean, we travel all over the world. You can travel anywhere you want to. I don't see any way, because Ernst Mayr is correct. You cannot form another species unless you separate it. So, I don't know, if we send people to Mars and they stay on Mars for some long period of time, maybe the Mars conditions may cause them to change, but I mean, who knows if that's going to ask Elon Musk. Maybe he can answer that question better than me. Or ask ISRO, because they can probably answer that question. I can't answer that question. But surely we are all one species, and there's no evidence that there's even a remote difference between us in any way. So not now, I can say.
Shannon, there are some online questions. Can I read
Please.
them out to you? Can being born in an apple influence the area in the brain which responds to an apple smell, so that the feature which separates the flies is something that's developed at birth rather than genetically?
So I never answer this during the talk, because I always know somebody's going to ask this question, okay? So yeah, this is a great question, and this is such an easy idea, right? So you grow up in something and then you like it as an adult. So easy, right? But no, it doesn't work in these flies. You can take hawthorn flies and you can rear them in apples. Now, we've only done this for about 3 or 4 generations, but that's like 3 or 4 years, so it takes a long time to do these experiments. But at least after that amount of time, if you have a hawthorn fly in apples, its children in apples, its children in apples, it still likes hawthorns. There's no evidence that the fruit itself is causing this variability. It seems to be something innate in the fly's genetics itself and not in the fruit, but that's a great question. So. For the neuroscience of insects, oh, well, I think there's an amazing, amazing book out there on the morphology of insects, and it's actually an arthropod morphology, and this is a really great book, and unfortunately I'm blanking on the author's name right now. No, Nick Strausfeld, thank you, it came to me, thank you. Nick Strausfeld. And it's an amazing book. It's so beautiful. He studies the morphology of insect brains, and he's even studied a lot of paleontological brains as well. So you can actually go back millions of years and look at all different kinds of brains, not just from insects, but crabs and lots of things. And I really would suggest that that they look at this book just because it's beautiful. And Nick Strausfeld's a really cool dude, so.
Excuse me. Hello. Yeah, so my question is basically, how come there was a complete opposite rewiring of the species in such a short amount of time? As in, since what we have seen, the evolution is a rather gradual process and mutation, but suddenly this, over a short period of time, this opposite thing seems rather counterintuitive.
Right. So these are not officially species. No current species definitions that we have would claim these to be different species, okay? So if you want to ask me what a species is, then it's a whole other talk and another course, and perhaps the rest of my career to discuss what a species is, because there's lots of definitions out there. The most commonly accepted one is mating, that you mate with each other and your progeny are not as successful, right? That's the common definition, the biological species concept. There's lots of concepts, okay? But by none of those concepts are these 2 different species. The idea is actually they're undergoing speciation. So actually we're watching the beginning of it, okay? They're not completely speciated yet. They're just undergoing it, and if all keeps going as it is, they will become different species. So they become the really great system to study for that reason. Because we're not studying something that maybe happened millions of years ago, but something that's happening right now as we can watch it. How it happened, there was a change in the brain. Why it happened is back to this question I can't answer to you, okay? Well, there's some sort of standing variation. That is what actually Darwin told us, right? Not everybody thinks the same, right? Even with us, we have lots of different preferences, lots of different choices we as humans make. It's not that stretch of imagination to imagine that insects are the same, and they have different preferences. And this must have been the case with these, and some of these preferences allowed them to exist on apples. So, but they're not different species yet.
Yeah, here to your left. Hi.
Oh,
Yeah.
hello.
So I was wondering what made you choose VR over, what made the transition to VR, to experiment on VR? And secondly, I was wondering, what does the apple tree benefit from all of this?
Oh, the apple doesn't benefit at all.
So then
right?
It should fit.
No, not necessarily. This is a pest, right? The apple doesn't, but the apple's job is to have some lovely bird or mammal eat it so that it can spread its seeds, right? That's the purpose of a fruit. A maggot in the fruit is screwing everything up. It's actually destroying the fruit and making it rotten so nobody wants to eat it. It's bad. So the fruit does not want this maggot around, okay? But the maggot does exist. This is an inherent, this is a famous fight. Plants and insects have been in a love-hate relationship for as long as they've existed, right? There's lots of insects that destroy plants. There's lots of insects for which the plants could not live without, like pollinators. So as far as I know, the fruit gets no benefit. We might be able to think of something over a beer, but I can't think of anything right now, okay? Now, VR. Why did VR? Is because I sat and watched these flies for many, many, many hours. My husband can attest to this, me sitting outside watching them and coming home very late at night because of this, okay? And I had these visions like, what if I could move the tree just over here to the left? What if I could take all the apples off this tree? What if all these apples suddenly became hawthorns? I had all these crazy imagination that it would be so nice to do, but of course you can't do them in reality. But with the dawn of VR, I can do it. I can do it in a virtual world. I can make the tree turn into something else. I can make it grow big or small, dynamically change it, and do a lot of really experiments that I would take months or years to do in the real world.
It would also be reproducible for other such experiments, for other insects, let's say.
Yes. In fact, that's the idea. So actually, we work on a number of different systems in our lab. We work on pests. We also work on pollinators. So we also have a love-hate relationship with our insects in our lab. Our other insects are mostly coming from India. They're insect pollinators like the giant Asian honeybee. We work on hoverflies, which are a great pollinator. We work on the coffee white-stem borer. That's it. This technology is not designed just for the applefly. I show you it for the applefly, but we are also even testing it on other insects. We just had hoverflies in it the other day. So.
Hello.
Hi. So I was just wondering whether in more complex brains, this phenomenon that you showed in which you alternate the two sites depending on whether you like them or not, the hawthorn site and the apple site, whether whether say among humans, someone likes jackfruit, someone doesn't hates this, whether it's the same kind of thing?
So I'm not sure about the switch part, but I can give you a couple examples with humans. So coriander, right, is actually a really interesting, has an interesting relationship with humans. Okay, most Indians will like coriander. I also like coriander, okay? But there's a mutation in the olfactory receptor gene. Okay, you guys know that when you eat something and you taste it, you're not really using your tongue. Most of the time you're using your nose, right? The flavor of your food is actually your nose. Taste is just sweet, salt, bitter, okay, umami, and maybe fat now, okay? There's not that many senses for taste. When we taste the flavors, that's actually the smell. And there's a mutation in one of these olfactory receptor genes that exists in about 10 to 20% of the population, particularly in Northern Europe, where the coriander suddenly tastes like soap. Soap, it's disgusting, okay? So, and that's because of this way it binds to the receptor has changed, so it does become a love-hate. So there is instances of this even in humans. So.
Hi, I have a question over here.
Way in the back, okay.
See, basically you're talking about the change in their behavior, like liking a fruit, not liking a fruit because of the change the
Yes.
neural activity.
Yes.
Is it possible to get this artificially, like to do that, to change change the behavior through artificial means?
Yeah, so theoretically that could be done in two different ways. Either you could change the wiring yourself through development, and I mentioned CRISPR as a technology that might be useful for that, so you actually change the wiring. Alternatively, we could actually, and what we actually would love to do with our virtual reality, and that's actually my dream, is to actually make like the Matrix, so that the flies actually aren't really in a world at all, but we just stimulate the regions of their brain that would be stimulated when they think about something, and then we can actually change them so they're actually going to apples, but they think they're going to hawthorns. Okay, that's really wild. That's what I want to do in the future.
And
So
another question. You told that you have been stimulating that in a virtual reality, right? How did you isolate the, the order of the fruit?
through a lot of chemistry, okay? And it was done a lot by the lab that I was in at Cornell. So what we do is we actually take the headspace around the fruit, so you can isolate the fruit either on the tree or you can pick it freshly. You can't use old fruit, right? These flies don't like fruit that's like laying on the ground. They like it in the tree. So you can take it and you can put, for example, a bag around it that you've cleaned, a plastic bag, and then you can take something called an adsorbent, which can be activated charcoal made from coconuts, which you use in your water filters, is excellent for this. And it actually can adsorb all the chemicals from that are coming off that fruit. And then you can use chromatography, gas chromatography, for example, to elute those volatiles and then isolate them. That tells you what is in the fruit. That doesn't tell you what these animals like. To do that, you have to combine it with other techniques like electrophysiology and behavior to then isolate which chemicals it is. It's a very long process, but it's something that we do also in our lab, so you can come if you want to see it in action. So.
Thank you.
Hello, I had this one question that you're usually using the dyes to measure the neurons that are responsible to carry out like a particular phenomena. Is it possible to know the electrical impulse or like the electrical signaling that is happening there and reciprocate it to artificially make like a pseudo prosthetics from these things, maybe like an extension, just by giving the same signal that you just got it?
Yes,
But
yes.
yeah.
So essentially what the brain is giving is electrical impulses, right? That's how the neurons work. So yes, we can record those. That was the gentleman's question here, that he was asking if we only used imaging. We also used electrophysiology, which you take an electrode and you can actually contact single neurons or groups of neurons in the brain and record their electrical activity. You can also stimulate those neurons by sending a current back into those neurons, and then you can artificially stimulate them, which would bring me back to the matrix, which is what I want to do. So yes, technically you can do that.
All right, thank you.
Hello, ma'am. I just had a question that as we saw here, that these flies have innately affinity towards the particular fruit they choose. So is there any case that these flies also have innate memory wherein they choose reject some particular fruits which are perhaps dangerous to them, evolutionary perspective, like?
Yeah, so these are specialists, okay? They only like these particular fruits. So lots of animals are not specialists. Bees are a good example. Bees pollinate flowers, but it's not like they only like a certain flower. They actually go to a wide variety of different flowers. And they have something called floral constancy, which means that they remember the flower and they return to it over and over again. These flies, you can't teach them to like something else. Like you can't teach an apple fly to like blueberries. You can keep giving it blueberries, keep giving it blueberries, keep giving it blueberries, it'll still go to apple. In fact, the only thing that happens when you keep presenting it with another fruit is that it hates it even more the next time and it still goes to the apples, okay? So you can't really teach them because of the way their brain has evolved. It's evolved to be specialized on this particular fruit. However, there's lots of systems and even lots of insects where you can train them. Bees are an excellent example. They live to learn. They learn so well, and you can teach them to like colors and shapes and smells and do all sorts of amazing things with them. But these flies, no. I mean, I'm not going to say that they're stupid, but you can't teach them to like new things. But they surely remember. They remember that they've been to this fruit, or they remember that they've been to this fruit that they hate. So.
Thank you.
Okay, if there is no other question, let's thank Shannon again for this wonderful, fascinating journey, for sharing this.